So this is about a on the surface, it's about a new group of insects that Rob Cannings and I and a bunch of other people worked on and discovered and named. But really behind it is there's a bunch of story about the beginnings of the modern world and what we can tell about that from British Columbia fossils of that age. So we named a new suborder of insects. I know, first of all, that not everybody's an entomologist, not everyone's a geologist. So I'll go through a little bit of terminology. I'll try not to get too much using odd terminology. Mostly insects are divided into orders. Those are the largest groups, like the beetles are in order, butterflies and moths are in order, grasshoppers, crickets, and katydids is in order. We're looking at an order called the Odonata, which is, as dragonflies and damselflies. Those are the two suborders of Odonata. But we discovered a third suborder in the Cenozoic, which we named the Cephalozygoptera. And that's kind of a big deal. I've named probably close to 100 species and maybe half that many genera, a handful of families, I don't know, 3 or 4, and one suborder, which people very, very rarely get to do. So that was kind of cool. So I'm going to tell you about these, about its discovery and about its implications. All right. Uh-oh. Let's see if this works. Oh, good. It does work. So what I'm going to be talking about, let's orient ourselves in time first. Now that we kind of have an idea about the organization of insects, a little bit of geology here. We're looking at the Cenozoic. And once again, you don't see all these things covering up Cenozoic, do you? No. Okay, that's good. All right, so the Cenozoic is the time period, but you do see my cursor moving around. Good. Down here is the extinction of the dinosaurs. For a long time, people had it at 65.5. Now we've redated it about 66 even. So down here is extinction of the dinosaurs. So it's really pretty, we're looking at a not long ago chunk of time. And up at the top, up at the top is us. That's humans in the Holocene. I guess we're going to call it the Anthropocene or something. Here is the ice ages, Pleistocene. These are not the relative links. Like, for example, the Pleistocene is almost 2 million years. The Eocene down here is a little over 20 million years. So these are not to scale. I'm interested in the Eocene, which is from the Greek, meaning dawn of the recent, and it's really aptly named. So I like looking at the early Eocene here, which is called Ypresian. That's named after Ypres in Belgium, where the famous, battle was. The famous, was that World War I and World War II? I think World War I.
One.
Yeah. Okay. And but there, the rocks of this age were defined and are very securely defined, and so we call rocks of that age anywhere in the world Ypresian. So I'm interested in a series in the Ypresian. There was tectonic uplift happening throughout southern British Columbia as continents and continental plates shifted. And all through from across about 1,000 kilometers from Driftwood Canyon Provincial Park up near Smithers is right there, down through Horsefly, southern BC, near Princeton at the Allenby Formation, and then Republic, there was high elevation. Down here, Vancouver, Victoria was still low elevation at this time. And if you can imagine a loaf of bread rising up in the oven, it gets all those little kind of cracks in the top. Well, that's what happens with uplift in the land as well. And you get what's called grabens, and there's downdrop blocks as the earth rises, and in those grabens can form lakes. And there's also volcanism, which may have dammed rivers, etc. So there's all these lakes formed throughout this region, and all of these lakes filled up with sediment in these uplands, and those sediments preserve a wonderful view of life at this time. So these sites are all around 51 million years ago. So if the extinction of the dinosaur is 66, that's not that long, a dozen or so million years after the extinction of the dinosaurs, when life has regained its diversity and bounced back. So let's take a look in here. Okay, up in north, there's Driftwood Canyon Provincial Park, and this is a wonderful site. You can see the shale here. This was a lake bottom filling up with mud, just all this organic matter just drifting down year by year and making these different layers and preserving the leaves and fish and insects and things that settled on this mud. And then eventually over the millions of years, compressing and lithifying and making the shale that we see and splits nicely upon those lines. And in central British Columbia, we see this famous locality near Cache Creek. And you can see the shales exposed in the hillside here above the Thompson River, and there's Highway 1. You can see a bunch of these. Hat Creek, I've collected quite a bit of amber there. Horsefly over at Falkland, at Quilshana. And then the southernmost of the series at Republic, just over the border in northern Washington. This is a wonderful site. You can go and visit it. There's an interpretive center there called the Stone Rose Interpretive Center. It's got wonderful collections, etc. And they have a very active society. And this is a fellow, Greg. Greg. Okay. You know, I'm a scientist. We're all scientists here. We're not like, you know, believe in the supernatural or anything like that. But I'll tell you something, there are some people who have got this spooky good luck at collecting, and you sit by them and they're just finding all the good stuff. And there it is by lunchtime, they got a stack of great stuff. And you're still like, I got to get something good. Greg has that talent, that preternatural ability magnetic ability to make the good fossils come up. So you want to sit next to him and see the good things, but it'll make you like, oh, why can't I find stuff? So at any rate, yeah, some people are like that. So at any rate, this preservation can be extremely fine-grained and extremely detailed. Here's a very tiny wasp. You can see 1/2 millimeter scale bar at the bottom. This is from Driftwood Canyon. You can see even the details of the tiny hairs on the membranes of the wings of this wasp. And let's zoom in closer. There's your 1/2 millimeter scale bar again, and you can see even further the details. Understanding this insect and seeing it is limited by the lenses of the microscope, not by the fossil. It's as good as amber. The only difference with amber is you can turn it around and see different angles, but this stuff is extraordinarily good. And in fact, we can get soft tissue preservation a lot of times. Here are tiny larvae also from Driftwood Canyon. These are each only a few millimeters long. These are very tiny, and you can see the soft tissue preservation there. I've worked on a green lacewing with Vladimir Makarkin, and we could see the soft tissue organs into the abdomen and see the male genital organs within this creature, 51 million years old. So this is stunningly good stuff. So for the last so many years, I've been motoring through the different orders of insects, doing a lot of work with Vladimir on the lacewings, that is the Neuroptera, and doing a bunch on scorpion flies and various different groups. And so at a certain point a few years back, I thought, well, it's time to start doing the Odonata, that order of dragonflies and damselflies. And so I contacted Rob Cannings, everybody's favorite odonatologist, and said, hey, let's do this. And Rob was kind enough to join the team. And we worked on the dragonflies. That's the suborder Anisoptera. And here are dragonflies. We had 9 fossils. And in this group, we figured we had 8 species and a handful of genera, and here are some of them. Almost all of them are in this group called the Aishnidae. That's the family, which is a modern family. And one of them was in this modern family called the Gomphidae. Now, these 2 families are very diverse today. They're not the most diverse, but they're very diverse. And these dragonflies would not look at all unfamiliar flying around next to a pond or a stream. You wouldn't think much of it, like, yeah, okay, there they are. And so we defined all of these, and then we put out a paper, I guess it was 2018 or so. All these papers are available on my website to download, almost all of them, brucearchibald.com if you want to go there. Then we turned to the next group, which we thought, oh, these are all damselflies, and it had something like 57, I think it was, specimens, a large number. And here's one from Republic, a very beautiful specimen. Now you can see from the dragonflies here that mostly what you get in fossil insects are the wings. And so I've learned quite a bit, as much as I can, about wing venation. And the wing venation is kind of like the Rosetta Stone. You can read who these things are and who they're related to, et cetera, by understanding the names of the wing, the names of the veins, and the different areas of the wing, and how these all relate to each other. So, so that's what I worked a lot with. But one of the nice things about working with Rob is he looks at modern ones, and so he's a lot more familiar with like the terminalia on the end of the abdomen and some of those other body characters that I don't really see too often in the fossils. So here we've got this beautiful specimen from Republic, and I want to understand the wings and the wing venation, but all of these, all four wings, the two forewings and the two hind wings are covering over each other. You can see there's bits of the body here. The head's gone, much of the abdomen's gone, and there's only bits of legs, but we got all four wings. So I imported this into Illustrator and worked on different layers and color coded each wing. And you can see for, you know, use the color code for hind wing and forewing over in the end there. And in that way, I could separate out, these are the same wings from that same insect. And once these are separated out, we know what's a forewing and what's a hind wing, et cetera. Then suddenly there's a whole lot we can do with this. And yes, it took a long time, but you know, tough luck. If it was easy, they'd be doing it in high school, right? So we figured this was a genus that had already been named, called Disagrion. And it was Disagrion is well known from another formation of the same age in Wyoming and Colorado. This genus is from Colorado, called the Green River Formation. I'll speak more about the Green River Formation a little bit later. And Disagrion is in an extinct family called the Disagrionidae. So that was kind of cool, understanding a new species of Disagrion, which kind of ties us to this, this mid-continental formation. And here's a Disagrion from the Green River Formation. Now, I should just say we were an upland, a temperate upland with mean annual temperatures about 10 to 13 degrees roughly. I'm talking very roughly here, in a very, very warm world. And I'll talk more about that later. So our mean annual temperatures in these lake bed sites, and by the way, I don't think I mentioned it, all these sites collectively are called the Okanagan Highlands. I should have said that earlier. So the mean annual temperatures of these sites was much like Vancouver or Victoria today. But you get down to the coast and the mean annual temperatures were probably more like Caracas. It was high atmospheric carbon at that time. So these Disagrion here from the Green River were from a lowland that had that hot temperature as well. So we found all sorts of Disagrionidae, and we named a bunch of new genera, well, a handful of new genera and many new species. Here's a hind wing of one, a beautiful one we called Okanagrion hobani. This is from Republic. This is a hind wing. And these specimens are absolutely gorgeous. Here's a forewing. We had lots of different specimens of this species, which was great. A lot of times you get in fossil insects, you get individual ones, and that's it. You got the one to work with. But with some of these, we had many specimens, so we can look at the group. And another one as well, another forewing. And not all of them are perfectly, are very complete or mostly complete. We get a lot of ones that are broken up. Now you imagine that these wings have to get into the lake, so they're going to die and land on the lake surface, float around for a while, and then make it down through the water column to this bottom mud. And in these, there's probably anoxic bottom, so there's not decay and there's no scavengers down. There's cold and low oxygen or anoxic bottom water. So these things just sit there like in a fridge until they get covered up in sediment. But some of them are going to get kind of wrecked up on the way. Some of them might have been imported by high energy streams into the lake and get in that way. And so they're going to be broken up, or there was scavenging, or there was all sorts of things could have happened to them. There is a subdiscipline of paleontology called taphonomy. And taphonomy studies all of the different things that happen between the time an organism is alive and the time it goes through fossilization and winds up in your lab table. So you got to understand taphonomic factors like floating time and all that before you can start to piece together an ancient community. And here's some more fragments of Oconagrion hobani in the Dysagrionidae. Now, they're not always just loose wings. Here's one where it's kind of fuzzy, but we do get the abdomen with the terminalia on here. We can see some of these terminal appendages, the male terminal appendages, the head's missing. But this is pretty good. And another thing we can't really see the venation, but we can see the color patterning. This is definitely hobani once again. And we can tell from this one, for example, that the forewings and the hind wings had the same color patterning, which isn't always the case in modern ones. Sometimes the fore and hind wings are quite different. So this one's telling us a lot. Here's a beautiful specimen from Republic with a different color patterning. You can see a smaller space there, and it's just different. It's a bunch of things and a bunch of detail. Not all of them are colored. This one had clear wings. Stenodiaphanus is another genus. This one from Republic. Now, when you split the rock, it will, in all of these sites, it will split evenly along the plane that was the bottom back when the lake was episodically being rained down upon by this mud. So it splits nicely along those planes, and you can get part of the fossil on this side called the part, and part of the fossil on this side called the counterpart. And here's the part and counterpart of the same wing. And so you can see here on this side, you got a lot of detail here that's missing here, but you got a lot of detail here that's missing here, and you got a lot of detail in the middle that's missing there. So when I'm doing these drawings, I do them from both the part and the counterpart, and they always look much better if you put a little ethanol on them. Everything looks better under the influence of alcohol. So I do these drawings dry and with ethanol, and from the part and the counterpart. And in that way, I can build up an accurate picture and figure out these veins. Now, it's not always so easy. The specimens are sometimes pretty scrappy, like this one. And a specimen like this one will take a lot of time and energy under the microscope to get together what you can. And we figured out that this is an Oconagrion of a particular species. And it's got all these landmarks here that we can use to identify it and define a species, etc. Now, this one shows something that we need to use from time to time in paleontology, and that is called parataxonomy. And I don't know how many of you will be familiar with parataxonomy. For this one, we could tell that it belongs to the family Dysagrionidae. We could tell that it's a different species than any of the other Dysagrionids that we've defined or know about. But we can't tell the genus of it because it lacks the character states that we need to understand the genus. So what we do is we place it in what's called a parataxon genus, and that's just like a holding place. So Dysagrionides is a parataxon. It doesn't have a diagnosis. It doesn't have a type species. It just has a definition. And the definition of Dysagrionides is all of those species which we can define as species that belong to Dysagrionidae, which we can't fit to genus. So we're figuring that as better specimens are known, this will go into either an existing genus or a new genus. We don't know which. So it's useful. Also, if let's say you're doing deep sea dredging and you come up with a bunch of larvae and you don't know what adults they go with, you might use a parataxon for that as well. Or let's say you're a botanist and you find a bunch of fossil wood that you can define to a species, but you don't know what leaves they go with. So you use a parataxon as well for that. All right, so now we get down to the mystery part. We started finding ones with heads on them. And this is where one day Rob Canning said to me, he said, look, Bruce, these heads just look a little bit weird to me. I'm not used to seeing heads that look like that on damselflies. These eyes, here's the head. These eyes are awfully close together and they're not bulging out. And the head is sort of a long front to back, and that's unusual. We don't see that in modern. By the way, this was named after Graham Beard, this species. We named it after Graham, who runs or did run a fossil museum up in Parksville and has contributed a lot to BC paleontology. A wonderful guy. So thank you, Graham, and it's an honor to name a species for you. So we started looking at these heads. And here's another one found by Rod Bartlett, who has that preternatural ability to find wonderful things, stacks of them, before you've even gotten out of the car. And he found this one, which is a beautiful one. Here we see the male terminalia on the end, which we could describe. But also there's a head up here preserved on this one. So we started looking a lot at the heads. And here's another one from that site near Cache Creek, which has a beautifully preserved head. And we're seeing the same thing over and over again with these heads that don't look at all like damselfly heads. Damselfly is the suborder Zygoptera, and we had assumed these were all Zygopterans, but now it's starting to look weird. Here is your standard Zygopteran head, your damselfly head. Here's a Lestid, family Lestidae, and you can see that always in damselflies, the head is short front to back, not counting these mouth parts, just front to back. This is a very short distance. The eyes bulge out like globes, and the space between them is about twice the width of one eye. So it's about twice the distance in here. But this is not at all the case in our insects, our new insects. Now, there are not a lot of character states that are diagnostic of the suborder as a whole that define it, but these head-shaped characters are important in defining the suborder. Now, there's also the secondary male genitalia have some details, but you're never going to see that in any of these fossils. Maybe if you're lucky to get one in amber, I don't know. So the heads are very important in understanding the suborder affinity of these insects. And the more we started looking then through the literature, about 150 years of literature, here's something that was insect described by Hagen in 1858 from Germany, I guess from Siblos. And it has this funny looking head that's not at all like a damselfly. And he wrote that the head was flattened. What is that? Kopf platt gedruckt. Excuse my pronunciation. So it turns out that people had described these insects for 150 years, and every time they saw one of these heads, they went, oh yeah, it was just squished somehow in fossilization. Over and over again, they said the same thing. But we noticed that it was always in either the family Disagronidae or this family, the Ceblosiidae, and they have very similar wings. So we thought, well, if this is the actual head shape, this is something new and different, and it's a big deal. Let's take a look at the taphonomic factors of fossilization and see if we can ascribe this head shape to some sort of pressures or things that happened during fossilization, as all these authors had assumed. So these were all lakes, like we see here, and the insects flew or blew out over the lake and landed on the surface and floated around for a while in the surface tension, and eventually made it, those that weren't consumed by frogs or scavengers made it down through the water column, and those that weren't consumed by fish or something landed on the bottom. The lake filled up with mud. Here's up at Cypress Bowl, just above me here in North Van, or I guess West Van. And all these male ants have had their nuptial flight the night before, done their business, impregnated the queens, and then died. It's a short and sweet life for a male ant. And they're going to float in the surface tension for a while. But they're not going to enter the fossil record. You can see that the bottom is you know, very shallow here, and they're going to probably get eaten up or decay in the bottom. If they're out in the middle of the lake, maybe this might be a fossil site in millions of years. Unfortunately, there's also a taphonomic bias, back to taphonomy again, and that is that insects with higher surface area to volume ratios float longer. So something like a damselfly or dragonfly has got big wings relative to its mass, and it's going to float and float and float, and probably the chances of it being eaten or decaying are very great. Whereas an insect like a beetle or a locust has got a lot of mass with not that big surface area. So it's going to break through the surface tension pretty fast and enter the fossil record. So your community is going to be, your fossil community that you find is going to be skewed in terms of this surface area to mass ratio thing. And you're going to see lots of beetles, less lacewings and damselflies. However, what we think is going on here, what I think is going on here, and other people seem to agree, is that what we're having here is diatom blooms. Diatoms entered the freshwater realm not long before these Okanagan Highlands sites, not long before the Eocene. And so it appears that they may have been forming sticky traps, basically at or near the surface, which would stabilize these insects and bring a higher ratio more close to the community composition of insects of varying surface areas. So we do see a high ratio of of these odonates and of lacewings and those kind of high surface area insects. And so we believe that this has brought this closer to the living community structure. So lots of things going on. All right, so what happens if an insect gets squished during fossilization? This is after it's entered the fossil record and the lithification process is taking place. It's starting to be turned into rock. And there's geological shear forces that actually squish the whole rock is kind of moved like that. And you can see here, this whole insect is squished along that plane. You can see one doesn't just affect the head, it affects everything. The wings are obviously changed, altered, the body, and also anything else like a leaf or a fish or anything else in these fossil beds are equally deformed in this way. So that's not what's happening with these heads that we're looking at, because here's one of those cebulliciids, and here's the head that is very odd looking. You can see the eyes are not bulging out, they're pressed to the head. It's about one eye's width between them. And this front to back is much longer than in the damselflies. So this is a classic head, exactly like what we're seeing. It's a cebulliciid from France, a little bit younger after the Eocene. And here's the same fossil, and you can see that the wings and the abdomen and all that are not distorted. So we can be confident that these heads were the actual head shapes in life. And all of these authors for 150 years were wrong. So this is starting to get to be a pretty exciting mystery. Here's that head shape, and to remind you, here's that Elestid, family Elestidae. Here's that shape here on this guy. You see the very wide area, the bulgy eyes, et cetera, et cetera. All right, so we decided to take the big leap. Must be bold. We took the big leap and named a new suborder, the Cephalozygoptera, which means head damselfly. Zygoptera is damselfly, and cephalo means head in Greek. And here are more Dysagrion specimens from the Green River of Colorado. And you can see that these are going to be this new Cephalozygoptera, and they're Dysagrionidae, and here's a regular damselfly. So the way they differ is that the head width here, let's just see if I can, okay, width of the head. Okay, width of the head is in here from the eyes, outside of the eyes, is about twice the length, twice the distance from the front to the back, not counting the mouth parts. Whereas in the damselflies, you can see that It's about 3 to 5 times wider there than it is here. So this is a much different basic shape to the head. Secondly, the compound eyes are more adpressed to the head in Cephalozygoptera, and like in that Cephalocyde you just saw. And here in a damselfly, they're bulging out, the globular shaped. And thirdly, the width of the eye in a damselfly, like I say, it's about twice an eye width here between the eyes, and here it's about the same as an eye width between them. So we've got a diagnosis here of this new suborder, which is strong. And we can't say anything about the secondary male genitalia because we don't know, and we may never know. But we did all of the phylogenetic work. And oh, I should say there was actually quite a team of us. It was Rob and I, and it was also Seth Bybee and his student, Robert Erickson at Brigham Young University in Utah. And they did the phylogenetic work, coded all of these character states and ran it through some software and came up with a tree, or a number of trees, which supported our idea that this is a new suborder. And Rolf Matthews from Simon Fraser University, who was a paleoecologist and was the 5th member of our team, the 5th beetle. He was so so the group of us worked on this for, I don't know, at least several years. All right, and so that takes care of, what is it, about 72 of the, no, 72 of the 78 specimens. We've now defined all into the Disagronidae and the Cephalozygoptera. And we also had to create a new diagnosis for the Disagronidae and look at the worldwide history of them and look at all the specimens from around the world, or high quality photographs of them. And we looked at the Cephalocydae as well, et cetera. And it took a long time, but that's fine. It was actually a lot of fun. So that's, what did I say, 72 out of 78, I think I said. So we found 5 specimens of these really weird ones, which weren't Disagronidae at all. It's like, what? And these are all from Republic, Washington. And they have some characters that are very odd, certainly don't look like Zygoptera, like damselflies. Now there's some technical things here, for entomology things. For example, this is called the arculus down here. And arculus is very rarely down near this cross vein called AX1. They're usually up here by AX2. This thing's pushed back. Also, this pterostigma here is very long and skinny and very unusual. So there are some things that set this thing apart as very unusual. But a lot of the rest of this wing looks like our Cephalozygopteran wings. Unfortunately, we don't have the heads for any of these guys. So we tentatively placed it in Cephalozygoptera, said this is what we think it most likely is, but we'll see, we'll confirm this once we get ones with heads. Might be. And here's another specimen. We had 5 specimens. Now, it was a really nice experience. We found this on the traditional territories of the Colville tribal people. And I've got some friends in that group. And I asked them, would the elders be interested in providing us with a word that we could use to name this genus? And because it's a new family, it would also be the family name. And the elders were happy to, they were enthused to be involved in this project. So it was a really nice interaction. And they provided us with a word in their language called huethuetax. And huethuetax means a dragonfly-like insect. So we called this genus Huethuetaxa, and the family is therefore called Huethuetaxidae. And so that's, there you go, a nice interaction. And so that gets us 77 specimens out of the 78. And the last one, the 78th specimen, is a damselfly, is Zygoptera. And it belongs to this family called the Euphaidae, which today is mostly, it's across a lot of Asia, but mostly in Southeast Asia, in hot parts of Southeast Asia, beautiful metallic colored wings. very pretty damselflies. This is from a subfamily of the Euphaidae, which is extinct, and the subfamily is very kind of odd. So here's the big picture of the Okanagan Highlands. We get the dragonflies are very normal looking. The dragonflies are kind of like what you'd expect to see by a pond today, and everything else in the order is weird. Even this damselfly is of an extinct subfamily, which is weird. So we have these major groups evolving or showing up at kind of different rates in here. So let's take a look at them around the world and let's place these in context of climate, geography, etc. The oldest one known is from the Cretaceous of China. The oldest Disagrionid is one specimen from China. So it was contemporaneous with the dinosaurs, just one guy. So now we're going to start their history in the Cenozoic. This is a time scale. Here's the extinction of dinosaurs here, about 66 million years. And then we get younger here, and it comes up to us up here in the Holocene. Now, these time units are the proper proportion. So you can see the Eocene is a huge chunk of time within the Cenozoic. This line here is a global mean annual temperature climate curve. So you can see how temperature across the Earth has changed from the extinction of the dinosaurs to us. Here we are in ice ages and all that kind of stuff. So what I'm interested in is the Eocene, this time period here, and this is very interesting. Here's the Eocene-Paleocene boundary. We see something happening here right at the boundary, and here at the end of the Eocene, we see another big event happening. We see something happening in the middle here as well. What's happening here is called the Paleocene-Eocene thermal maximum, or the PETM. This is becoming very well studied. And a lot of this is done through, this climate analysis is done through cores, sediment cores drilled offshore, where you get this finely gradated record. But what happened here is suddenly there's this thing called the carbon isotope excursion. There was tremendous amount of carbon dumped into the atmosphere and the ocean. We figure, or they figure, people who've worked on this have figured there was several thousand gigatons of carbon dumped into the atmosphere within a short period of time, like 10,000 years or something like that. These numbers keep changing because people are still working on it, but along that scale. So geologically, a flash. And this pumped up temperatures, so that I've seen recent estimates of sea surface temperatures in Denmark being like 30 degrees mean annual temperature. If you can imagine that, just boom, the world gets hot. And then we can see a decline as this carbon is reabsorbed back into the system. And the whole thing might have been a couple 100,000 years. Although, like I say, those numbers are likely going to shift and change. So with a super huge event like this, you probably saw last summer the UN Committee on Climate Change said what a disaster it would be if we got past 1.5 degree increase in global temperatures. Now, if we got to 2 degrees, we'd be in a whole lot of trouble. I've seen estimates of the PETM, a minimum about 5 degree jump and up as high as a 10 degree jump. Yeah, I should let that sink in for a minute and imagine what that would be like. People are looking, there's a bunch of money being pumped into PETM studies because it is analogous to what we're doing today if we don't watch out and clean up our act. So you see lots of extinctions happening at this time, lots of new things showing up later after. So it's a time of great change, and also range changes of animals and plants, as you might well imagine, as different parts of the globe have different temperatures than what you realize. Here, this is the Okanagan Highlands time. This is the Apresian, the early Eocene. This is what we're seeing called the early Eocene climatic optimum, and there's a bunch of more carbon in the atmosphere than we have today. And there's a lot of ways we can tell atmospheric carbon levels. For example, leaves have stomata in them, the little windows that let gases in and out, and it's a trade-off between too many of them and you lose moisture, et cetera, and too few of them, you're not getting enough gases through. So there's a trade-off and a balance depending on the amount of carbon in the atmosphere. So you can look at stomatal density and give an estimate, and there's all different ways to do this. So we go through a decline in world global mean annual temperatures. This area deserves more study, but I'm not aware of people doing a whole lot with this. Right here, that has received a lot of attention, though. This is called, and excuse my French, it's in French, it's called a Grande Coupure. Any French-speaking person, please forgive me for that. In other words, the Great Cut. There's a sudden drop of temperature here and a big turnover, for example, in Europe of mammals, et cetera, although there may be other factors involved. This marks the end of what we call the global greenhouse climate. Greenhouse climates go way back to the Paleozoic, so hundreds of millions of years ago. And from here to now, we've got what we call the global icehouse climate. Here, we had very equable, this is going to be important and play into this. We had equable climate, so the summer to winter temperature difference was very small, whereas today we got, cold winters and hot summers here. And we had a low temperature gradient, pole to equator, all sorts of things like that. Here at the end, we probably had increase in pole to equator temperatures. We probably had increased seasonality, summer to winter, and all these events happened here. For example, Australia pulled away from Antarctica enough tectonically to allow the circumpolar Antarctic current to sequester cold waters and the heat transport from the equator to the Antarctica changed, and all sorts of other things happened. The ocean became layered. Before here, we didn't have the same layering of the ocean. So all of these different events happened, and suddenly we had this big change into the Oligocene. So that's important to remember. All right, so let's see how this affects our insect communities. Oh yeah, Paleocene, Eocene, okay. So let's get down to community ecology. Here in the Okanagan Highlands, we've got this weird tropical temperate mix of insects. We've got a bunch of temperate sort of insects like this snakefly, which today almost all live in temperate northern climates, and another group of sawflies, which are very boreal as well. And that's what you'd expect in a cooler upland northern community. You have all of these things, and also plants as well. So you got spruce and things you expect to see in one of our forests. But it's mixed, this strange, weird mixture with tropical insects and plants. You get it's a tropical beetle-like cockroach, a Diploptera, which is native of Fiji, but it's been obviously, we spread it all over the tropics with shipping and all that. And here's one from the Cache Creek fossil site, which is a Diplopterine, again, and you get all these other things which today only live in the tropics. And in the forest structures, you had palms, a palm spruce forest, palms and spruce living together in the same community. So what's going on? What's going on with this? And what we figure is that once you raise the winter temperatures, that allows a whole lot of things that live in the tropics today to come up into colder areas. And many of those things don't require hot temperature. They just don't like cold winters. And in that way, you get a temperate place like these Okanagan Highland uplands, which had, 10, 13 degree mean annual temperatures, but you didn't have frost days in the winter. So tropical things are happy to mix at this time. So what this means is that we had tropical lineages in the Eocene ranging well high. Like I say, in the lowlands, it'd be hot, but in any uplands, they would have them as well. And then later, probably after that Grande Coupure at the Eocene-Oligocene boundary and then going into our time, tropical lineages filtering out and going down to lower latitudes. So you'd had this sifting going on later in the Cenozoic. Now, I did my dissertation on diversity, on biodiversity and climate. And what I did was I took a sample in the lowland tropical rainforest of Central America of insects. I collected a large sample, and I took a sample in a temperate zone of Massachusetts, where I was going to university, where I did my PhD. And I compared those two insect communities with the Maccabee site up near Cache Creek, the Okanagan Highlands site. So the tropics had high mean annual temperature and low seasonality. The temperate zone one in Massachusetts had high seasonality, but low mean annual temperature. And the fossil site in the Okanagan Highlands had low seasonality and low temperature. And so I went through all this stuff and compared the diversities of these, and found that the high diversity was associated with seasonal stability, with temperature stability. And so it's looking like what happens here is that we had high diversity. Now, at this point, we also see high diversity in temperate sites for the same age in Patagonia and South America, et cetera. So it looks like high diversity existed well outside of the tropics, well towards the poles. I don't know about the extremes, but way outside of where they have today. And now high diversity is restricted to the lower latitudes. And so it's what I'm calling the pear-shaped model of Cenozoic biodiversity. So we started out the Cenozoic with this big extinction event, killed the dinosaurs. Then we have this bounce back and a high diversity through the end of the Eocene. And then after that, end of the greenhouse world, we get into the icehouse world, and global diversity is attenuated. And here we are, we're killing everything off. So good for us. So that's what I figured global diversity looked like over the last 66 million years. Oh, another thing I should just mention before I'm moving on about diversity. Some of you may be familiar with the famous paper by Dan Jansen called Why Mountain Passes Are Higher in the Tropics. And he explains why equable climates promote a bigger turnover from valley to valley in mountainous regions of the tropics than they do in the temperate zone. That's another whole story. If you ask me back sometime, I'll tell you that story. It's called beta diversity, the diversity across the landscape. And in a study I did here at Simon Fraser, we found that this had a high diversity turnover from site to site across the Okanagan Highlands as well. So this held up, this whole idea of different levels of diversity as being high in the early Eocene, the uplands of British Columbia. Okay, so one other thing I wanted to tell you about these communities is that I'm surprised these insect communities that I've been collecting have been very little work done on them. And so it was sort of like the golden age of discovery. There's been a little bit of work. Mark Wilson from the University of Alberta in Edmonton did some work in the 70s, and a couple of people did some in the early 20th century, but not a lot. So I extensively collected and went to a lot of museum collections and looked at tremendous numbers of these. And I've been surprised to find the numbers of insect groups, which are very ancient, which I kind of figured would probably have died out by then. Like these particular kind of stick insects, which are known from the Jurassic. And here we find them again in the Eocene. And a number of other groups as well that are ancient and survived and then aren't known after this time. So the big extinction event end of the Cretaceous didn't kill them off. A second group of insects are groups that appear in the Eocene and were gone by the end of the Eocene or shortly after, maybe in the Oligocene, et cetera, the Miocene. But there's a whole group which are only like this very weird scorpion fly here called Holcorpidae. And the third group is, of course, groups which either originate in the Eocene and then become a big deal today, or originated back in the Cretaceous, but were a tiny little element in the communities and then suddenly blossom out and become a big deal and in the Eocene to our modern day. And ants are a good representation of that. Let me just read a few fun facts here for you. Ants are about 15 to 20% of terrestrial animal biomass, maybe 25% of animal biomass in the tropics. A single colony in the tropics was found to eat 21,000 caterpillars per day. Single hectare, 8 million individual ants, et cetera, et cetera. Ants are incredibly important in ecosystems. and go deep into the Cretaceous, but they're almost nothing until boom, around Okanagan Highland times, here they start to become modern. We see a lot of the dawn of the modern world, old things ending, overlapping with new things beginning, and then a chunk in the middle, which are just Eocene. Getting more and more interesting. Here's another group which goes back further, controversially into the Cretaceous, although we don't know if that fossil's really that old. A couple of specimens in the Paleocene of France just before this. And then we start to see them blossom out in the early Eocene, and with a good record, not many, but still a good record here in the Okanagan Highlands. Bees, flowering plants, obviously. So biogeography. At this is a figure that I made for a 2011 publication, and it's changed since then, but this will do for our purposes. Europe and North America were not separated as far by the Atlantic then, and you could walk from the Okanagan Highlands onto Greenland, through forest all the way without getting your feet wet, onto Northern Europe, down through Scandinavia, there's Spain and all that. Or you could walk down through Greenland, through the Faroe Islands and the UK, down onto continental Europe, or across the Bering, what would become the Bering Strait, into East Asia. Now, this is the North Sea, and we now know that the North Sea was never completely enclosed, that one or the other of these areas were open, but that, whatever. The point is that there was a free flow of animals and plants at this time in the early Eocene. We have the greatest similarity of mammals between Europe and North America of any time at this time. There were times when there were palm trees growing right up to the Arctic Ocean. The Arctic Ocean was at times enclosed with a fresh water on the top and a bunch of azolla water plants growing on top of the Arctic Ocean. So we had this great interchange happening. We had the PETM had just happened and bumped everything around. Lots of things going on. So let's just look at a little bit of the history of the Cephalozygoptera. Here we have them at these fossil sites here in the early Eocene of North America, and here in the Green River, the lowland hot site, here upland temperate site, and then later in the Eocene, right before that big crash in the end of the greenhouse world, over here at Florissant, Colorado, beautiful interpretive center there. And after that, after the end of the Eocene, the whole Cephalozygoptera is not known in the Western world. If we go to Europe, here we are right after that PETM event. And I'm pleased to say that I've been contacted by some of the people in Denmark working on some of these odonates from there, and we're collaborating on them. And we've discovered the Huehuetaxidae in Denmark, here in the Eocene of Denmark. So that's papers. We just got the page proofs accepted today. So that paper should be out any day now. These sites, amber, Baltic amber, and over here, this is right at the end of the Eocene, right when there was that crash, just before that big crash. And here's about the same age as the Okanagan Highlands. This represents some over in Pacific coastal Russia. We get into the Oligocene. We don't see the Disagronidae anymore. We just see this Cebulosaeidae throughout this region. The first time we see them. Then we get into the Miocene up here, which is shorter than that's shown on this. And we see the end of the Cephalozygoptera. This is the youngest here in this area called the Tortonian, so about 10 million years old. And that's the end. The Cephalozygoptera is gone. We got a couple of scrappy Disagrionid fossils, but mostly Cebulosaids near the end of the Miocene. So what's next? Rob Cannings and I just worked on another project with some new fossils. All of these what I 78 or whatever fossil, plus the 9 dragonflies. Those had built up over many decades and sitting in museum drawers as well. So we're not going to get a big chunk of them again like that. But new ones do come up all the time, like this one from Princeton. And this looks pretty chewed up, but we can work with it. And we have this thing in review right now. So I mentioned the discipline of taphonomy, one of the different subdisciplines of paleontology. And the last thing I'll tell you about is another subdiscipline of paleontology, and that is called ichnology. And ichnology is the study of fossil traces of animals. It can be their trackways, or it can be their nests, or their feeding damage, or any of those sorts of things. If you didn't know what made these tracks, you could compare them with trackways in different parts of the world and say, oh yeah, the same species would have been in France at this time or something, you know. And so there are people who are dedicated to ichnology. One of the fellows is Conrad Labandiera at the Smithsonian, who the bunch of us are working with right now, a whole group of us, to look at more about these odonates. Oh, come on, there we go. Odonates lay their eggs, not all, but some odonates lay their eggs in this fashion on leaves. And we've got a number of leaves from these sites. And so we're working with Conrad and his associate, Jorge Santiago Blay, on these leaves with oviposition scars. And we can tell if these were laid on living ones or dead ones, because the living ones would have had reaction tissue, you know, healing tissue. The dead ones floating on the water would not, obviously. And we've got a couple of botanists working with us, Kathleen Pegg and Melanie DeVore, identifying all these leaves. And Rolf Matthews is paleoecologist. We're going to look at how they lived and put together the story based upon these ichnofossils. So there's a lot left to do. This has been a long project, and hopefully we get it done this year. Who knows? And what else have I got to say? Well, here's some more eggs on leaves. That's it. That's the story. So this Cephalosygopter paper and a bunch of other ones are all at my website. And brucearchibald.com or brucearchibald.org, either one will work. And you can download them for free from there. And here are all the people who are working on these projects or are working on current projects. Robert Erickson, and Seth Bybee, Rolf Matthews, Conrad Labandiera, Jorge Santiago Blay, Kathleen Pegg, and Melanie DeVore. That's been the crew, and that's the story. Thank you.